So let's just celebrate this moment, which is historic for that we are in person and remote. Beautiful. This is our new life. So it's so incredible to see all these faces of these collaborators who want to hear you, Anthony. And we won't have time for everyone to introduce themselves, but we have Cliff Sarin from UC Davis. We have Philippe Golden from UC Davis, who's in Sweden. We have Olin Wolkowitz, who you know well, who I think connected us in the first place. And we have amazing staff postdocs and faculty. So feel free to be asking questions in the chat, and I think Elena can field the questions to you, Anthony, so you don't have to monitor the chat. So Anthony is a phenomenal colleague who does basic and translational research. He studied in Greece with George Crusoes. Has anyone heard of George Crusoes? One of the fathers of stress research. And then he did a postdoc at Max Planck in Germany. And now he is at Duke as an assistant professor. And he is just doing amazing discovery-oriented research, drilling down from patterns of epigenomic correlates of psychopathology to functional, to seeing if they matter functionally with his cellular studies. So I don't know anyone like you, Anthony, and we're interested in everything you do. So share with us what you would like. I'll just mention that we, Eric led this, we collaborated with Anthony on our data on SAGE. And this was a caregiver study, and he did some, I don't know if you're going to mention it today, but
I will.
Okay, then I won't go there. But now we are working with Anthony on data on the Black and white cohort we've been following since they were 10, and looking at lifespan stress and methylation patterns on stress-related genes, led by Agus and Elisa, and some of our other postdocs are joining the wave of excitement as we discover what we can discover here. So welcome, Anthony.
Thanks so much for the kind introduction and welcome. And what a nice group to be discussing with, I should say, today. Yeah, let me share my screen. I remember there was this nice time that we met in San Francisco in person. I wish we could do that, but still it's very
Napa.
nice.
Was it Napa, Owen, with Cindy?
Yeah, yes,
Cindy.
Napa,
Yeah.
that's right.
Yeah, we will do it again. Okay, we can see your slides, so this is perfect.
Slides, great, okay. It's always confusing with the Zoom. Okay, so I just want to mention I was before at Duke, but now I'm at UNC. Our lab is studying as already mentioned by Lisa, we're studying the intersection between stress and aging-related disease. And our approach is to use data from large-scale human cohorts and cells, where we mechanistically dissect the associations we find in humans. And we use both computational and bench work approaches. And with that, I just want to acknowledge already the heroes of our lab that have been able to continue this work during the pandemic. And this is a picture we took back in May when we were able to go back to in-person meetings for a dinner. So with that, I would like to start and let's see. Okay, just to take you through an outline. So I will just mention background and motivation for doing this kind of work. And then I would like to take you through 3 research teams that are ongoing in our lab. Psychosocial stress and epigenetic aging. And I know many of you know about this, epigenetic aging. You have some ongoing work. Stress-driven programming of inflammation. And then how we study prolonged stress in cells and the epigenetic at cell states resulting from stress. And then finish with some future directions and the take-home message. So background, in terms of background and motivation, I have been interested to do this work for a very long time. And actually it is embedded in the Greek culture, but also in other cultures. The idea that if we stress too much, we will age before our time, we will die before our time. This is something that my grandma used to say when I was little. And that's something else that you hear people say, that diseases result from too much stress. We just try to do much more than we can. And there are many, many examples of characters in literature, movies, and so on that appear to age very fast when they're exposed to high levels of stress. On the other side, individuals, and this is a legendary character, Chinese, who was able to control his stress levels, lead a peaceful life, and that made him live for a very long time. So there's this general idea that if we live fast, then we also die young. And living fast meaning we have high levels of stress, excessive stress that is maladaptive. And this was brought in biological terms by one of the fathers of stress research, and Hans Selye, who said that every stress leaves an indelible scar, and the organism pays for its survival after a stressful situation by becoming a little older. And one can say that it is interesting to study whether these indelible scars are epigenetic in nature. Just to emphasize this by giving you an example, in Victor Hugo's Les Miserables, Hugo says that it was he, Jean Valjean, the main character of the novel. In fact, the clerk's lamp illuminated his countenance. He was pale and he trembled slightly. His hair, which had still been gray at his arrival to the court, was now entirely white. It had turned white during the hour he had sat there. So again, this idea, if we're exposed to high levels of stress, we have this accelerated aging, which is used in the literature to emphasize that the stressor is very intense. And to many of us that do some clinical work, we know that we see these phenotypes of accelerated aging, in individuals that are exposed to stress. But for some others, it might seem more of a science fiction, so to speak. But intriguingly, there was this paper relatively recent that indeed showed that there is a mechanism, there's a molecular mechanism that can explain graying of hair and accelerated aging. And this was a study done in mice. So in terms of an epidemiological evidence linking stress and age-related disease, there are multiple studies showing that stress at different points in time, early in life, cumulative stress, but also stress-related phenotypes such as PTSD are associated with accelerated aging. And our lab and others are interested in what are the molecular and epigenetic mechanisms linking these conditions. And as I will mention as well, we're very interested in inflammation because this appears to be a key mechanism explaining stress-related disease risk. So in this context, I think the stress response and the stress system provides a very powerful mechanistic framework. And when we talk of the stress system, we can conceptualize it, starting it at the level of the limbic system. When we have a stressful event detected at that level, we have this neurohormonal cascade. HPA axis is not the only axis, but it is the most well-studied probably in this context. So this neurohormonal cascade culminates in peripheral secretion of glucocorticoids, the main glucocorticoid in humans being cortisol. And I just want to point out that there's this negative feedback that homeostatically regulate the HPA axis. I will get back to that a little bit later in the talk. And this is actually part of the study that we did with Lisa and Arik, where we studied this. And what is really interesting is that cortisol is very lipophilic or hydrophobic, if you prefer, traverses essentially all of our cells. And once it goes inside the cells, it activates the glucocorticoid receptor, which is a transcription factor primarily. And the GR binds to different sites of the genome, where we have these glucocorticoid response elements, but also through other complex mechanisms, this binding happens. And when this binding occurs, we have changes in the transcription of genes. It has been estimated about 20% of our genes respond to glucocorticoids. At the same time, it has been shown that there might be molecular memory that happens at the site of binding. So it's sort of a remodeling of the epigenome. And this memory serves for the cell to respond differently. That's what it is told to happen in the future when the cells are exposed again to stress or glucocorticoids. So in terms of epigenetic mechanisms, there are multiple mechanisms and they act in concert. None of them acts in isolation. Nevertheless, we are focusing primarily on DNA methylation, and in particular the so-called CpG context, CpG meaning the cytosine guanine dinucleotide. The reason for this focus, one of the big reasons for this focus is that this is one of the most, perhaps the most well-studied mechanism in humans. And this is thanks to the advent of DNA methylation arrays a few years back. So we'll be talking a lot about this mechanism. Now, all of these questions are very important. We all know about the aging crisis. We know that individuals worldwide age more and more. It is estimated that in 2050, we will have, for example, tripling of the individuals above the age of 85. At the same time, we have more and more reports of higher stress levels in Western societies. And this is not even taking into account the stress of COVID. For example, during the COVID pandemic, it has been estimated that there is higher levels of stress and anxiety disorders to a level of 30 to 40% increase. And of course, all of this is very important because AIDS is the single most important risk factor for several diseases that are leading cause of morbidity and mortality. And this includes cardiovascular disease, for example, Alzheimer's disease, and so on. And so we have to really be prepared for these conditions, for this upcoming challenge for health systems. So with that, I want to start with the themes. I don't know if we want to wait until the end, or feel free to interrupt me. I don't have I think the slides will not take more than 40 minutes, so we should have time. If you have something, a nagging question, feel free to interrupt me along the way. So in terms of epigenetic aging, so you know, it's epigenetic aging or epigenetic clocks, you will find it in the literature. It's the same idea like any other measure of biological age. It started by an effort to predict chronological age, meaning what is the age of on one's passport. That's what we mean by chronological age, passport or ID, whatever you prefer. And there are these biological measures that are trying to predict this age of the individual. And DNA methylation age turns out to be a very accurate one, the most accurate we have. So in an ideal world, we would have a perfect prediction between the two, chronological and DNA methylation or biological age. But we know that some individuals appear to age slow, biological as compared to their chronological age. And one popular example is Sean Connery, who within these 20 years showed no signs of biological aging. On the other side, there are individuals that appear to age very fast. And one popular example is Barack Obama, who within the 8 years of his presidency showed clear signs of accelerated aging. And the idea that has been set forth is that if we subtract chronological from DNA methylation age, we get this epigenetic age acceleration metric, or delta age, which has been shown, and this is some of the earlier studies, but this has been replicated by many other studies, mostly. So epigenetic age acceleration has been shown to predict or be associated with multiple age-related diseases, including cancer, cardiovascular disease, all-cause mortality, and so on. So it does appear to be a promising marker of aging, which is the reason why we have been focusing on it. So to just show you how this works, in case you're not familiar with the marker, you see that when you have DNA methylation age, so you know, to get to this measure, you just need blood or any other tissue DNA. And then you have to combine multiple methylation sites that are scattered across the genome. In this case, we used one of the earlier markers, which is called Horvath. It was developed by Steve Horvath. And you can see how just by knowing the DNA methylation status of the epigenome, you can have this nice prediction, which has correlation coefficients usually exceeding 90%. At the same time, you see that many individuals fall above or below this line. And what we wanted to answer back when I was doing my PhD was whether this variance can be explained by exposure to life stress. And indeed, we found this association between life stress and delta age. This is cumulative lifetime stress. higher levels of stress was associated with accelerated epigenetic aging. And this association was only observed in older individuals, not in younger ones, even after adjusting for the total levels of stress. And at the same time, it was dependent on the type of stress. So it was only the personal life stress, not the network life stress, meaning the stress that affects only the social network of the individual. So the personal life stress appeared to drive this association. But we went a little bit beyond these associations, and we had some data of individuals that received an oral synthetic glucocorticoid called dexamethasone. And we had methylation levels before and after. This is all data in blood, by the way. And we wanted to see if the sites that comprise the Horvath epigenetic clock, which is 353 sites, by the way, methylation sites, we wanted to see if those sites respond to glucocorticoid exposure. And indeed, we saw that about 1/3 of the sites were acutely responsive to glucocorticoids. Most of them were demethylated, which was interesting to us because at the site of glucocorticoid, of GR binding to the genome, you actually have acutely, as a more immediate response, decrease in methylation. And also, we noticed that the closest that these sites happen to be to a glucocorticoid response element, the more likely they were to become significant in showing a response to glucocorticoids. Everything above this dotted line, I should have mentioned that, is significant. So every dot is a single CPG site, and so everything that goes beyond this line shows significant changes in response to glucocorticoids. So some of our ongoing studies after I started my lab at UNC, following on this work, the study I showed you earlier was in 400 individuals, and now we are conducting a study using data from the Women's Health Initiative in 4,000 women. Interestingly, in these women, we don't see an association with Horvath clock, and this is again with stressful life events, but we do see an association with Grimm age, and this is something that is based on the Hannum clock, but a little bit different. It's called extrinsic epigenetic age acceleration. So these are sort of, especially the Grimm age is a next generation epigenetic aging marker that is more predictive of mortality instead of chronological age. And another study we have going on is, there is this study which is called Aurora, and it consists of individuals that present to the emergency room, and they're followed over time. And so we have neuropsychiatric outcomes in these individuals, brain aging and epigenetic aging. And we find very interesting associations again with, actually this is a prediction of outcome after exposure to trauma, which is very interesting, in association with Grimm age. And these are collaborations. This work is led in the Women's Health Initiative by Harleen Skinner, who is a postdoc affiliated with my lab. And this is in the Aurora collaborations ongoing with Sam McLean, Sarah Lindstedt, Kerry Ressler, and Karstan Kennan. And by the way, we are also extending now this, where we plan to extend, I'm planning to put a grant together to extend epigenetic analysis, but to also measure telomeres. And this will be collaboration with Elisa and Ju that I hope to make progress with, and my hope is to submit it in June, but we'll see. So with that, I want to move to the next theme. Again, feel free to interrupt me if there were any questions. So the next, another very central theme that we're studying is how inflammatory states can be programmed by exposure to stress. And this work again started when I was doing my PhD. I was interested in this molecule called FKBP5. The reason we were interested in this is that FKBP5 is part of the glucocorticoid receptor complex in the cytoplasm. And actually, when FKBP5 is bound to the GR, you have decreased translocation of the GR to the nucleus. So you have decreased activation of the GR essentially. But when the GR gets activated, actually, FKB5 is one of the most glucocorticoid responsive genes, and probably this is explained by the multiple GREs that are scattered across the gene locus. So what this does in effect is when you have GR activation, you have higher FKB5 levels. As I mentioned, this has an inhibitory effect on GR signaling. So in effect, this creates this ultra short negative feedback loop that homeostatically regulates GR signaling inside the cells. Interestingly, FKB5 is the most glucocorticoid responsive gene. If you rank all the genes in human blood in terms of their transcriptional regulation, FKB5 will be ranked first. So for these reasons, I was interested to see how epigenetic regulation of KB5 might be driven by both aging and stress, or what might be the interplay between aging and stress. And these are data from about 3,000 individuals from different cohorts. This is the Grady Trauma Project. This is a German cohort called CORA. And this is another cohort we had at the Max Planck Institute of Psychiatry in Germany. But actually, I have seen the same pattern in 6 or 7 cohorts by now. So there is this very consistent decrease in methylation that happens with increasing age. That has to do with 2 methylation sites that are in the promoter of the FKB5 gene. So those 2 sites are age regulated, but at the same time, those sites appear to show an accelerated decrease with aging in individuals that also have symptoms of depression, but also I'm not showing here early life trauma. So there seemed to be, just to summarize this, an epigenetic regulation at the level of these 2 methylation sites that are in the promoter of the gene. And this was through an interplay of aging and stress. Now, interestingly, this was also associated with an upregulation of FKB5. And at the genome-wide level, this was with data using gene expression arrays at the time. We could see that higher FKB5 levels was associated with a signature of inflammation-related genes, and in particular, genes that appear to be targets of NF-κB. So the whole message was that there was an epigenetic upregulation of FKB5 by aging and stress, and this was contributing, we thought, to NF-κB-driven inflammation. And because inflammation is such a risk factor for cardiovascular disease, we also extended these findings, and we looked at whether this same signature is present in individuals with a history of myocardial infarction, and indeed that was the case. We only had data from 2 cohorts. And we indeed saw that there was a decrease in methylation at the same 2 sites in individuals with a history of MI. So what are some follow-up of this? And this is the study that I mentioned earlier with Elisa, that we conducted with Elisa and Arik, that Elisa was very kind to share the data with us. And what we looked at here is the so-called post-dexamethasone suppression cortisol levels. So the dexamethasone suppression, maybe you're more familiar with that than I am, or at least some of you. But the way this works is you give a synthetic glucocorticoid, dexamethasone. And as I mentioned, there is this negative feedback loop. So you expect that the system will shut down, the HP axis will shut down when you give dex. So if you have higher levels of cortisol after exposure to dex, this suggests that you have a dysregulation of the HPA axis. So in this case, higher levels of cortisol after dex were associated in the data we had in the SAGE cohort with decreased methylation of TNF, suggesting a pro-inflammatory epigenetic signature. Another study that we recently published was using data from TSST, or the Trier Social Stress Test, in case you're familiar with the test. So in this case, individuals are asked to present in front of the committee. They also do some calculations. And we had data of exposure of IL-6 levels before and after exposure to stress. And interestingly, individuals that showed higher levels of IL-6 responsivity, they also had a decrease in vascular function over time, and an increase in metabolic resistance, in insulin resistance, which is a marker of metabolic dysregulation over time. This is data, longitudinal data over 12 months. We are, just to give you some examples of how we model this and how we corroborate these findings with cell models. This is again back in the study I showed you with FKP5. So we had all these associations in the human cohorts, and we wanted to see if we can recapitulate these findings in cells. So we use this T cell line called Jurkat cells. We overexpressed FKP5. This is just a control plot showing the overexpression worked of FKP5. You have higher levels after overexpression. And in cells that had overexpressed FKP5, we indeed saw a higher NF-kappa B signaling and higher secretion of IL-8, which is a key chemokine that we decided to study. There are some other details that I skipped, but this suggested that indeed we see this pro-inflammatory state when we overexpress FKP5 in cells, corroborating the mechanism. And again, from data that Elisa and Ju and Arik shared with us, we found that when we have higher levels of TNF methylation in the SAGE cohort again, this was, by the way, ex vivo data. So these were immune stimulated cells, and then there was a DNA that was isolated from these cells. This was mRNA isolated from these cells. And we correlated this with methylation signatures that we have in the same individuals that participated in the ex vivo study. And we saw that lower methylation levels of TNF were associated with higher TNF expression after stimulation, so functionally supporting this epigenetic signature. And more recently, we have been interested to go beyond glucocorticoids, and we have been, so we do expose cells to cortisol, but we also use norepinephrine as a model. Interesting with norepinephrine, you see this acute pro-inflammatory signature happening, and this is in a monocyte cell line. At the same time, if you pulse cells and you withdraw exposure to norepinephrine, you see this molecular memory of a pro-inflammatory state, so which persists even after the withdrawal. So some follow-up studies of that. I have been fortunate at UNC to work with a group of epidemiologists that are part of the TopMed consortium. And this is Eric Witzel, Gerardo Heiss, and Laura Raphael. And the TopMed is a large consortium of studies that are funded by the NHLBI. And we have estimated access to more than 23,000 individuals from this consortium. So we have been studying how stress is associated with methylation signatures and cardiometabolic data in these cohorts. At the same time, all of this is whole blood data, right? But I have been very intrigued to go beyond the mixed cell signature to see if we can deconvolute these into individual cell types. And through collaboration with Aran Halperin, who is a brilliant computational biologist and computer scientist at UCLA, we're trying to see if we can deconvolute. And intriguingly, there are ways now to do this computationally. So you just have whole blood data, and as long as you know the cell type proportions in your sample, which you can again calculate using methylation data, there are computational ways to say, for example, in CD4 cells, we have these methylation levels predicted computationally, and then we can see if this predicts or mediates risk for cardiovascular risk. And this, I think, also being able to computationally deconvolute in specific cell types will enable us to more precisely dissect the mechanism. And this is in the lab work that has been led by Calvin Liang, who is a postdoc in my lab. And for this, actually, we recently received an R1. So this was my first R1. I was very, very excited a couple of weeks ago to hear the news. So yeah, with that, I want to move to the last theme, just to show you a little bit on how we study epigenetic and cell states of prolonged stress. And we try to do this in a more naturalistic way, and I will try to explain what this means. Well, again, for sure, this has some limitations. But the idea is that stress can happen at different points in life. Usually individuals that are exposed to high levels of stress early in life also continue to have a high risk for subsequent stress exposure. And we hypothesized a few years back that this can act cumulatively at the epigenetic level. And we were trying to find a way to study this at the cellular level. And we decided to use fibroblasts, just because fibroblasts are an easy cell to culture, and it has the advantage that you can culture the cells for a long period of time. And we did the data I'm showing here is over the course of 7 weeks of treatment with cortisol. And when we say naturalistic, we mean that the levels of cortisol are actually those that can be reached in vivo when individuals are exposed to stress. And one reason that we did that is that previous studies have mostly used synthetic glucocorticoids or high levels of glucocorticoids, so we wanted to make it more physiological. Now, what was very intriguing is that if you track these cells over the course of time, so this is cells that are early in passage, and these two groups are cells either treated with vehicle or cortisol for 3 weeks, and these are cells that are treated for 7 weeks, and you do a principal component analysis of the methylation state of the epigenomic states, you see that these epigenomic changes accumulate over time, even over the course of 7 weeks. So the cells separate over time at the epigenomic level. At the same time, those sites that show this cumulative signature appear to be found more commonly in open C sites as opposed to CPG islands. They are found, as someone would expect, that more commonly they are enriched for GR binding sites and at chromatin accessible sites. So suggesting, a genomic context component of these effects. Interestingly enough, the fibroblasts also show very robust cell phenotypes. So cortisol in this cell line, fibroblast cell line, greatly promotes proliferation and extends the replicative lifespan. And actually, when I started using fibroblasts, I was expecting that I would see a pro-senescent phenotype, but to the contrary of my expectation, it was actually a pro-proliferative and even an extension of replicative lifespan. At the same time, we see increase in migration with cortisol, again at the physiological levels. And what was also interesting here is that we used a selective GR antagonist, and we found that these effects of cortisol are completely mediated by GR activation. And these cell phenotypes are corroborated nicely by transcriptomic data, which show that we have multiple genes. It's about, if I remember correctly, the number close to 1,000 genes that are changing over time in their level of transcription. And those are enriched for GO terms such as cell migration, adhesion, and proliferation, which makes sense with the observed phenotypes. Again, the sites that are regulated by glucocorticoid receptor activation are enriched for specific genomic contexts, including localization of proliferation and migration genes, again corroborating the observed phenotypes. So just to give you an example also of going beyond the epigenome-wide level, we are using some example genes to study how site-specific epigenetic regulation can give us fundamental insights. And this is a volcano plot of cells that have been exposed for 7 weeks to physiological cortisol levels, stress hormone levels. And we focused on this gene called ZBDB16. It's a non-glucocorticoid responsive gene, but it showed, in our data, it showed a very robust regulation. It was actually the most glucocorticoid responsive gene in this context. And you can see how this gene is literally turned on by exposure to glucocorticoids. It has 0 levels of expression without exposure to glucocorticoids. So it's really, cortisol opens the gates for this gene to become transcribed. So we thought it's a very nice model to look at what's happening at this gene. And using EPIC-DNA methylation array data, we found this very robust hypermethylation of the gene body. At the same time, there was a hypomethylation near the promoter of the gene. So this is a very intriguing, I think, and novel epigenetic signature that we're following up. So yeah, and I want to mention, and this is, there's this seminal work by Elisa, of course, started with this PNAS paper back in 2004, and also Owen has showed the same signature with depression, and this has been meta-analyzed. It has been shown to replicate this association with telomere short of stress exposure with telomere shortening. We are not heavily focused on telomere shortening, but we had this cell model, and we were wondering if exposure to cortisol in this context might be associated with telomere shortening in these cells. Again, to our surprise, we didn't see that. We found that there was no effect in telomere shortening. This is just a control of FKB5 showing that we have a robust genomic response with both cortisol and dexamethasone. Dexamethasone is is a stronger glucocorticoid receptor activator. In these cells, you see this decrease in telomere length as they are passaged over time, which is to be expected. So going back to the Hayflick limit. But at the same time, there was no effect on cortisol. And I mean, it is just that it is this particular cell type. We concluded that in vivo, there are most likely much more complex mechanisms happening, and perhaps depending on different cell contexts. So following up on this work, so this is Oksana, she's my lab manager, and she's following on how prolonged stress can influence epigenetic states. At the same time, I have been collaborating with some amazing epigeneticists at UNC. Nathan Hathaway, Brian Strahl, and Stephen Fry. We have been looking at how we can make it more mechanistic. For example, we have ways of doing targeted DNA methylation editing in the cells. This is, for example, Nate's expertise. And at the same time, we're extending this to fibroblasts that are coming from individuals, so primary cells, in collaboration with Trevor Archer at the NIHS. So some future directions and the take-home message. So, I guess my vision is to go, so as I mentioned, we're doing cells and human cohorts at this time, but really my vision is to get to that point of mechanistic translation where you have all the levels of translation, going also to model organisms. Mice are one example, but there are so many other model organisms when someone is thinking of aging. And for that, I'm starting to collaborate with some people at UNC and beyond. So one, for example, idea is to do stress epigenetics across species using human cohorts and cells, as I mentioned, but also model organisms like rodents. And someone can use potentially translatable biomarkers like epigenetic clocks. Epigenetic clocks are a very nice example in this context because, as I showed you earlier, we have this a nice prediction of chronological age using DNA methylation. But intriguingly, we have the exact same phenomenon in mice, but not only. Now it has been shown in so many species. Actually, Steve Horvath is now fully focused on, almost fully focused on doing it in different species. And there's this, even though the sites are not entirely conserved, for example, between mice and humans, you have about 50% of the sites being conserved, methylation sites. Nevertheless, you have the same exact idea and phenomenon, where you can predict very nicely the age of the mouse or the animal by using DNA methylation data. And so we're really trying to elucidate conserved mediators and pathways of stress and aging-related disease. And this is collaboration with Jose Rodriguez-Romaguera at UNC. And we just started the Carolina Stress Initiative, which is a translational initiative in our Department of Psychiatry. So, I mean, as a broader vision, I also like to think that one day we will have biomarkers of disease states using epigenetic signatures. This is one of the early papers in the lab of Elizabeth Binder, who was my mentor at the Max Planck Institute of Psychiatry, where they looked at whether PTSD can be distinguished into different endophenotypes. And in this case, it was whether someone has been also exposed to childhood maltreatment or not. There are, of course, many examples of using epigenetic signatures as biomarkers in cancer research. And we're also very interested in using epigenetic age in association with other aging markers. I mentioned telomere length, but also neuroimaging, where there are also now markers of neuroimaging that can predict. This is the same idea, they can predict brain aging. And even going further, the idea of biomarkers is with the one way we might be able to prevent or reverse stress-driven epigenetic signatures and associated phenotypes. These are just some examples in early life with epigenetic clocks. In these studies, they were targeting kids that were exposed to discrimination, stress, for example. They saw that the association between stress and epigenetic aging when the families were educated was going away. And there are a few studies in animal models showing that we can treat animals that are exposed to stress, for example, with glucocorticoid receptor antagonists, and we can prevent epigenetic changes and associated phenotypes from happening. Okay, and yeah, going back to this idea, of we have this chronological age again with biological age. Can we one day, using these markers, be able to identify vulnerable individuals like Barack Obama during the time of his presidency and stop the aging process, as it seems to have happened over the 5 years when he stopped being the president? Or on the other side, can we identify resilient individuals and use this knowledge to bring positive changes and a higher level of functioning? So, and with that, I want to finish with one last slide. So, for me, epigenetics takes us away from these concepts of DNA is our destiny, especially when we talk about complex phenotypes, and away from clear distinctions between nature and nurture, to ideas actually that I think are very interesting, that we can potentially change our nature by nurturing ourselves. And this goes back to ancient philosophers as well. Aristotle said that habit is second nature, and Heraclitus said that day by day what we choose, think, and do is who we become. So with that, I want to close and just acknowledge the different collaborations and the individuals who did a lot of the work that I showed you. And yeah, I'm happy to discuss further and answer any questions that might come up. Thank you. Let me stop sharing so that I can see you better, but we can go back to the slides if we need to.
Thank you so much, Anthony. That was absolutely amazing. That scope, as promised, from, molecule to macro, societal, and life experience. And so there are a lot of questions. I know I have a lot. I know everyone here probably has a question. And are there some questions in the chat? Okay, so why don't we just have the people online first go ahead and ask questions. Owen, did you have a question?
I did. Thank you, Anthony. That was great. And it looks like your current and future work's great as well. One of your slides mentioned induced neurons as one of the tissues you'll be looking at. Could you explain what you're thinking about there? I'd like some questions about that.
Yeah, that's very early. We're not even close, but we have this access to the primary fibroblasts. And there are two ways to do this. One can go back to the induced pluripotent stem cell state and reprogram the cells all the way to neurons. Or there are now ways to do that directly to neurons. There are protocols that someone can go directly from the fibroblast state to the neuron state. And there are advantages and disadvantages with this approach. If you go the way back to the iPSC, you are losing the epigenetic signatures. So you are more studying the genetic background. If you do the induced cell approach, you retain some of the states, not perfect, but you retain some of the epigenetic signatures. Actually, we are more interested in this second approach. That's why I mentioned that in the slide.
I agree. I think that's a really good way to go. When I was thinking that you were proposing doing the iPSC derived grid, I was thinking that given that that erases the epigenetic changes, might you then apply cortisol to those neurons from stressed and non-stressed individuals and see if there's a differential effect of cortisol on the epigenetic mechanisms in the people
Absolutely.
that, yeah.
Absolutely. Yeah.
Yeah. Okay. Thank you.
Of course, very complex approach, but yeah, absolutely.
So Anthony, there were so many amazing specific findings, and I'm wondering, are there generalizations that you can make about, for example, we know the methylation accelerates with age, less genetic and more patterned by life experience. How does stress work, traumatic stress, and how does glucocorticoids work? It didn't look unidirectional that we could always say, oh, they're increasing methylation of the inflammatory related genes. It looked like it's much more complex.
Yeah, you're totally right. It's really much more complex. It's context dependent. Some sites you see increased methylation, some other sites decreased methylation. It also depends on the duration of exposure, dosing. All of these things are important. For example, with our more naturalistic approach, we see about 60% after prolonged treatment with cortisol, 60% going up of the sites with epic array, 40% going down. And again, this is dependent on, for example, we see this site, gene body hypermethylation, but you might see in many cases in the promoter sites or enhancer sites decrease in methylation.
Thank you.
Looks like Maytal has a question.
We can't see everyone in the gallery and who's raising their hand. We only see Maytal. So Maytal, go ahead.
Oh, hi.
But I think Elena, if you could call on people after that, that would be great.
Yeah, thank you so much for this interesting talk. Relatedly to what Elisa asked, do we think more about these changes as like transient or more of long lasting? And besides, if this depends on like the duration of the stress, does it also depend on the timing when it happened, the timing of development, and about the reversibility? Yeah, like, is it depends, if it happened at like a sensitive time in life, or actually if it happened earlier because you have more room to intervention and more of like positive experiences to happen. We think that maybe, yeah, I don't know, just like this, just general questions about the reversibility aspect of it.
Yeah, absolutely. I can tell you what we know, or what we think we know, because unfortunately, we don't have answers to these questions yet about the dynamics of these changes. There are some early studies. I mentioned a little bit, 2001 study that was done in cells. So the duration of the exposure does seem to be important and this was done with synthetic glucocorticoids. So how well that speaks to the physiological condition, we don't know. But the duration does seem to play a role. So for these cells, if I remember correctly, you needed to expose them for 4 days to see a persistence over time. And then they checked again the cells after several weeks and the DNA methylation signature persisted. This was only in one locus that was done. And we plan to do it at the genome-wide level with our model after withdrawing the cells at the 3-week state. That's an experiment we're planning if we get funded to do it. And then I don't think we have a good answer with human studies, how they persist. I mean, there are these studies with early life trauma and the associations that someone can speculate that indeed those signatures persist. But I don't think there are good longitudinal studies that can have shown that unequivocally.
Thank you very much.
Sure.
I had a question. I noticed in your work you focus a lot on the HPA axis. I'm wondering, like the variability in epigenetic aging that is related to either like lifetime stress exposure or recent chronic stress exposure or acute stress responsiveness is the variability in epigenetic aging that you're interested in studying. Like how much of that do you think is mediated through the HPA axis as opposed to the sympathetic nervous system?
Yeah, as far as I know, no one has studied an association with the sympathetic nervous system with epigenetic aging. Also, as far as I know, no one has studied with prolonged exposure to glucocorticoids and epigenetic age. I might be wrong, but I cannot recall of any studies. So I don't think we know the answer. I can only say that you know, HPA axis is one very nice conceptual and mechanistic framework we have. GR signaling has been studied for many years. It's a well-studied system. Definitely not the only one. As you said, the sympathodrenal system, as I mentioned, we do some studies with using norepinephrine. Again, it's very difficult, all of this with limitations, like also when you model, we recognize the limitations that we have, you know, when you have prolonged exposure to glucocorticoids, that doesn't entirely capture what's happening in real life, where you have circadian rhythmicity and ultradian rhythmicity of glucocorticoid exposure. That's definitely a very valid question. How much of that is through this axis or the other axis, and hopefully we'll be able to answer that.
Well, because that Nature paper with the mice, right? Wasn't that implicating the sympathetic nervous system?
That's right. That's right. This was in particular, looking at hair and melanocyte stem cells that might be driving. Yeah, you're right. Yeah, so, you know, some can say that some tissues are more sensitive to one effector or the other. Even some individuals might be differentially affected.
Thanks.
Raise your hand if you have a question. Elisa, go ahead.
Mine's kind of small.
So again, wanting to understand general principles and simplify. So I think you showed us that glucocorticoid exposure increased overall methylation. And so I'm wondering, you also showed us that it's increasing, you know, cell proliferation and longevity, or at least telomere length. It's not shortening it. So the question is simply this: how do you see an inflection point from acute to chronic stress? Were you seeing an effect of hormetic stress exposure when you saw the improvement, cortisol and improvement in cell longevity? The duration of exposure in these experimental studies is obviously so important for whether we're going to see, at least in the telomere or telomerase, going up or down and telomeres lengthening or not. And we think it's a hormetic stress, like in the short run when the mice are, and Annalise Beery showed that when the mice are stressed for 3 months, they're healthy young mice, their telomerase goes up. In a predator model, telomeres lengthened in a different state. So there's this, adaptive stress response where telomerase becomes defensive. But the neuroplasticity literature shows cortisol is essential. Like in the strabismus model, you give rats this optic defect. If you want them to rewire normally, they have to have cortisol. You block cortisol and you don't get the plasticity. You don't get the repair. And so is that through epigenetics? Do we understand that, that kind of positive role of cortisol in plasticity?
Yeah, I mean, it has to be epigenetic at some level, because you have some, if you have remodeling, if you have plasticity happening, there are some molecular changes, some transcriptional changes that last in time. So it has to be epigenetic. Again, I don't think we know well how much of that is acute and adaptive versus, there are some few studies showing that acute and chronic exposure to stress might have very different molecular changes happening. I think there were a couple of studies by Bruce McEwen and But this was not looking at DNA methylation. If I recall, it was histone methylation studies. I do think that the signatures are very different when you compare acute and chronic changes. Many of the acute changes are reversible. There are some studies showing that if you expose to glucocorticoids, you have a reversible change that lasts 3 hours, but then goes away after 24 hours. Yeah, and I totally agree with your point that, you know, we don't want to erase stress, right? Stress is necessary for survival. We want these changes, but we want cortisol to, increases to happen. That's part of an adaptive response and survival response. But when it becomes maladaptive and chronic and persistent, that's where the issue is. And that's what we try to study with this model, with the prolonged stress. And actually, we use cortisol levels at the higher end of physiological stress.
And you think of that as a chronic toxic stress paradigm?
Yes, yes. You know, it is a question. It's like you have these changes. Are they maladaptive or actually good for the cells? You know, they proliferate more, they migrate more. So it really depends on the context. I can see that if someone applies that to cancer research, it's negative. You know, you have cells that proliferate more and maybe they give rise to metastasis by migrating more. On the other side, if you think of wound healing, it's actually good to have cells that proliferate and migrate. So that's an open question and limitation.
Yes,
for now.
thank you. Great, great answer.
Hi, Anthony.
Hi.
I have two things I'm
That's
curious.
Lisa. I don't think you can. see
Yeah,
her.
well, we've met. I don't know if you see me, but first of all, have there been any moderating effects of antidepressants on this effect between either induced or, you know, naturalistic stress and the changes in methylation? I'm just really curious about any kind of like protective factors that might, you know, lead to changes or prevent the demethylation or what you're seeing.
We haven't looked at antidepressants. We didn't have data to do that. I know Owen might have some very interesting data with antidepressant treatment. Yeah, unfortunately, we haven't looked at that. But interestingly, the only thing that came up when in this paper back in 2015 in Genome Biology, early life trauma was protective. And,
Early trauma was protective.
yeah, I was actually surprised by that. Initially, I started looking at that with the
So
idea.
oh, sorry.
Do we need to stop? I think we are.
I'm so sorry, I didn't mean to interrupt you. We have one more question from Victor Ruth. I don't know if you can see Victor, but Ethan's going to read it, and then we'll have to end. This has been incredible. Thank you.
Sure.
Yeah. Victor asked in the chat, have you looked at the heritability of any aspect of the epigenetic changes?
We haven't. All we did was to look at principal components, so adjust for the principal component, you know, of ancestry background. That didn't seem to change the results at all. But yeah, no, we haven't looked at heritability.
Victor, did you want to add any thoughts there?
No, thank you. That was it was a broad question because it's you know, currently a subject of some dispute, and there was some suggestive data that there may be some aspects that are heritable and others that are not. So I was just curious if anybody's looked at that in their data set, or in the animal studies also, you know, that you've talked about.
Mm-hmm. Yeah, definitely. We have some GWAS data in the Aurora study that we didn't have in the graded trauma project, for example, back then, but we do have in the Aurora. So yeah, it would be interesting to include this.
Yeah, and I'll just say Anthony is so creative. In our 2-generation data of moms and offsprings, he's looking at heritability of stress or SES-related CPG sites. So no one's done it that way before, and we'll have to stay tuned to hear the final results of that. But it does look like there are some sites that are transmitted. So I'm sorry to end there. You can put your questions in the chat if Anthony has a few moments afterward, but we hope to have you in person and we can have, easier discussions, but this was incredible. Thank you everyone for coming.
Thank you. Thank you for having me. Nice to see everyone.